Everyone, Dr. Beena here. Today I am going to revise the topic extracellular matrix and the disorders associated with it. The topic was taken recently and it was the new topic added in our syllabus this year. So we don't have any previous questions of university, but some of the important questions we can discuss in this class. So the extracellular matrix is the area between the cells. Important questions are composition of extracellular matrix, describe the structure, synthesis and functions of collagen, list the disease resulting from the abnormalities of collagen synthesis and maturation, explain the structure and functions of elastin and keratin, and one disorder, Duchenne Muscular dystrophy. So these are the one which I am going to discuss today, but you have to read the chapter thoroughly because anything can be asked from that chapter. Now, what is extracellular matrix? Extracellular space is filled with gel-like material called the extracellular matrix. It is the ground substance which provides structural and biochemical support for the surrounding tissue. So, it is a non-cellular component of all tissues and organs. There are three types of molecules in the extracellular matrix. The structural proteins consisting of collagen and elastin, and the proteoglycans, that is the protein-polysaccharide complex to embed the structural proteins within it. And then the third one is the adhesive glycoprotein to attach the cell to the matrix, that is fibronectins and the laminins. So here you can see the proteoglycans in which the fibrous proteins are embedded, that is collagen, elastin, etc. Then there is adhesive glycoproteins, that is fibronectin, laminin, etc., which are attaching the cell surface to the matrix. Now, what are the components of the extracellular matrix? The structural proteins consist of collagen, elastin, and fibrillin, and the specialized proteins, fibronectin and laminin. Then, of course, the proteoglycans, which consist of glycosaminoglycans attached to the matrix proteins. And you know what is the glycosaminoglycans. You have studied in detail. Then fibrous proteins are present in this amorphous ground substance containing water and minerals. So it contains water, minerals also. And now we have to explain the functions of extracellular matrix. There are two types of extracellular matrix, basement membrane type and the interstitial matrix. So mainly they are divided into two groups. Now what are the functions? It holds the cells together, provides porous pathway for the diffusion of nutrients and oxygen, and it protects the organs and provides elasticity to the blood vessels, lung and the skin. So it acting as a protective substance where the and also it provides elasticity to the blood vessels that it can stretch, dilate and contract. Then the lungs also expand and contract. The skin elasticity also is done. Then glycosaminoglycans and proteins form a gel-like structure, provides the flexible mechanical support, and it is acting as a lubricant against the shock, mechanical shock. Then it regulates the cell growth, migration and differentiation, and it is supporting growth and wound healing, especially for the bone growth. Now what are proteoglycans? Few words about proteoglycans. Major protein of the extracellular matrix composed of glycosaminoglycan chain covalently linked to the specific protein core. There are three types: leucine-rich proteoglycan, cell surface proteoglycan, which is acting as co-receptors for the signal transduction pathway. Then the modular proteoglycans, which mediates the cell adhesion, migration and proliferation. So proteoglycans is glycosaminin attached to the specific proteins. Now the structure and functions of collagen. What is collagen? It's the main protein of the connective tissue. Collagen forms around 25 to 30% of the total body weight of protein. Whatever protein in the body is present, out of which 25 to 30% is constituted by the collagen. It has got a tensile strength and holds the cells in discrete units. So it holds the cells in discrete units. It is present in almost all organs like bone, teeth, tendon, cartilage and blood vessels. So it is present in almost all the tissues. Now we'll pass on to the structure. Collagen is the most common glycoprotein in the extracellular matrix. There are three types of structures shown here. This is the one backbone structure or triple helix, and the other one is the wire diagram and the space filling diagram. And all collagens have a triple helical structure. That is the basic structure. Basic unit is the tropocollagen, consisting of 3 peptide chains, that is 3 alpha chains, each consisting of 1000 amino acids. 3 chains are twisted around each other to form the triple helix with which forming great tensile strength. The chains are held together by hydrogen bonds between the chains. Then there are multiple forms in the body. Around 19 distinct collagens are found, and its synthesis occurs in the fibroblast intracellular. Now, collagen has an unusual composition, amino acid composition, with around 33% glycine, 10% proline, and 10% hydroxyproline, and 1% of hydroxylysine. And 4-hydroxyproline and 3-hydroxyproline and 5-hydroxylysine is also seen fairly in large amount, but tryptophan and cysteine are absent, and tyrosine is less in collagen. So usually tryptophan and cysteine are absent in the collagen. Now the structure of collagen. Primary structure of collagen is, as you know, it is a polypeptide chain, and it's a regular arrangement of glycine, proline, or hydroxyproline. That is why it is written here as glycine, and X and Y may be proline and hydroxyproline. The glycine is every third amino acid of the polypeptide chain is constituted by the glycine. It being a smallest amino acid of all, can fit into the crowded interior and produces a groove, so that to which the other polypeptide chain can intertwine. So that is the importance of glycine. It can fit into a curve where other polypeptide chain can intertwine while forming the triple helix. Then what is the alpha chain? Alpha chain forms a left-handed helix, no interchain hydrogen bonds, because the nitrogen of the proline and hydroxyproline has no substituted hydrogen for the hydrogen bond formation. So that is why there is no hydrogen bond in the alpha chain. Now you can see in this diagram, the synthesis occurs in the nucleus, mRNA is going, and then it enters the endoplasmic reticulum, where the ribosomes forms the polypeptide chain, and it is finally enters the Golgi apparatus, where a triple helical structure is formed with the amino acid sequence at the amino and the carboxy terminal site. So after that, it is secreted into the extracellular space, where the cleavage occurs in the polypeptide on both sides, and finally a triple helical structure tropocollagen is formed, and then it is arranged in the quarter-staggered manner, and then forming the fibrils. So these are the fibrils which are interlinked by the bonds. Here there is cross-linking while forming the fibrils. So this is the overall synthesis part. Now the 3 alpha chain wound around to form the right-handed helix by forming interchain hydrogen bonds between the glycine amide of the one peptide with the carbonyl group of the adjacent peptide, and thus stabilizes the triple helix. Then after that, formation of collagen fibrils that is spontaneously aggregate to form the fibril. They are arranged themselves to form the quarter-staggered manner, parallel and overlapping areas. So before that, it is again the peptidases cleaves the amino acids on the both sides, that is amino terminal and the carboxy terminal. Each tropocollagen molecule overlaps the neighbor by the length of 3/4 of the molecule. So extracellular maturation occurs. Procollagen molecules are secreted from the fibroblast that we have seen in the previous diagram. It is secreted, and then it is cleaved by specific peptidases. There is an enzyme, specific peptidases, at amino terminal and 150 amino acid, and carboxy terminal and 300 amino acid are removed and to form the mature collagen fibril. That is, you can see here. So here this, and there is amino acid terminal and the carboxyl terminal. These are cleaved by the peptidases, and then it is caught a stagger arrangement occurs. That is the fibril formation or the collagen fiber formation. So there will be a cross-linking of the collagen fibrils, and then one condition called dermatopraxis is the deficiency of peptidase enzyme. This enzyme is deficient, so the skin is prone to torn easily. So it may torn easily whenever there is some injury. It will be breaking easily because both ends there are amino acid which has to be removed by the peptidases to form the mature collagen fiber. That is absent in this dermatopraxis. So here again you can see the cotostaggered arrangement of the fibrils, and then finally each fibril joins to form the bundle of collagen fibers. Now how the collagen is degraded. Collagen is degraded by collagenase enzyme produced by Clostridium histolyticum, a pathogen producing gas gangrene. Adult humans do not have much collagenase activity. Usually there is not much, but it will be acting in certain conditions like degradation of bone and cartilage resorptions, and then osteoporosis, tumor metastasis, postpartum involution of uterus, Paget's disease, rickets, osteoarthritis, scurvy, and rheumatoid arthritis. These are the conditions where the degradation of collagen occurs by the collagenase enzyme. You know what is postpartum involution? After delivery, already during the pregnancy, uterus is dilated. Then after delivery, postpartum period, it is involuted to the normal size. There again the collagen is acting and finally producing the normal uterus. Similarly, for bone resorption and lung tumor metastasis also. Now we'll pass on to the disorders associated with extracellular matrix proteins. So many are there, and few only I am discussing. You have to refer the other also. Mutation in the gene that is responsible for the production of collagen, that is producing osteogenesis imperfecta, EHLERS-DANLOS syndrome, epidermolysis imperfecta. These are the three important disorders where the mutation of the gene responsible for the production of collagen is occurring. When it is occurring, these diseases are produced. Then mutation of the gene coding for fibrillin produces MARFAN syndrome, and deficiency of the enzyme degrading the proteoglycans. We have seen proteoglycans in the matrix, no? So if there is deficiency of the enzyme in the degradation of proteoglycan, that will produce a group of disorders called the mucopolysaccharidosis that you have studied in your chemistry part. You have to refer back and study the mucopolysaccharidosis. Now we'll pass on to the osteogenesis imperfecta. It is also called the brittle bone syndrome. That is, bones are easily broken down. So this condition called brittle bone syndrome. Multiple genetic defects in the synthesis of type 1 collagen. Here the type 1 collagen is affected. There are many types of collagen depending upon the biochemical function and the mechanical function of the tissue. So that is type 1, 2, 3, 4, 5 like that. Many of the mutations are single base, and the substitution that replaces glycine in the repeat sequence by another bulkier amino acids like cysteine, which will preventing the correct folding of the triple helix. So we have already discussed how the triple helix are formed, where the glycine forms the curve. But if the glycine is replaced by bulkier amino acids like cysteine, what happens is there will be not forming a proper collagen. So the fibrillar array cannot be formed, resulting in the brittle bone. So the tensile strength is due to the fibrillar array of the collagen fibers. If it is not occurring, there will be chances of breaking the bone. That is, brittle bone syndrome occurs. The clinical features of fragile bone, thin skin, abnormal teeth, and weak tendons. These are the important features. And abnormally thin and translucent sclera produces a blue color due to the deficiency of the connective tissue. You know what is the sclera part, and through the sclera you can see a bluish tinge. In these cases, osteogenesis imperfecta. So that is again a typical feature of this disease. Now what is Cellular-Danlos syndrome? formerly known as cutis hyperelastica, a group of inherited disorder of connective tissue that is collagen, and it is caused by genetic defects in the proteins involved in the synthesis of and assembly to form the collagen type 1, type 3 and type 5. So these are the important area where the defects are notified. This is due to the defective lysyl oxidase or lysyl hydroxylase. So these are the two enzymes which is needed for the collagen synthesis. So then if there is no lysyl oxidase or this thing, then there will be forming of the type 1, type 3 and type 5 are not formed. And the clinical features are hyperextensibility of the skin and increased joints mobility, abnormal tissue fragility. So these are the important features of this disease. Joints are hyperextensible, can extend to any type, then increase mobility, joint can be mobile, and then also abnormal tissue fragility. Then epidermolysis bullosa, it's a rare disorder due to the mutation of the gene encoding, again collagen, characterized by skin breaks and blistering of the skin and the epithelial tissue. Blistering means a bullous-like lesion may be present on the skin surface. This disease is of three types: epidermolysis bullosa, that is three types of epidermolysis bullosa. One is dystrophic form, mutation of the gene coding type 7 collagen, which anchor the basal lamina to collagen fibrils in the dermis. Then junctional form, detect in laminin which leads to blistering in the dermal epidermal junction. So there is defective laminin formation, so it produces blistering in the dermal and epidermal junction. Then the simplex form, where the mutation is in the keratin filaments, which results in the blistering of the epidermis. So there are three types, that is dystrophic form, then the junctional form and the simplex form. Now there are other disorders, Menkes disease, where the deficiency of copper, defective function of lysyl oxidase, reduced cross-linking of the collagen. So for the collagen cross-linking, this is needed. Then deficiency of ascorbic acid, defective hydroxylation of collagen, weak collagen is formed, fragile blood vessels and poor wound healing. When there is fragile blood vessels, there will be bleeding gums, swollen bleeding gums will be there in the scurvy, that is deficiency of ascorbic acid. Then homocystinuria also you have studied, and accumulated homocysteine in the blood block the cross-linking of the by reacting with the lysyl aldehyde, produces skeletal deformity, vascular and ocular defects. So you know what are the skeletal deformities. Then there is ocular, that is subluxation of lens, lens zonules, there will be collagen which will be not functioning properly. So then that is due to the accumulated homocysteine, which blocks the cross-linking reaction by the acting on the with the lysyl aldehyde. Then laterism is another condition where the ingestion of toxic substance in Lathyrus sativa inhibit the lysyl oxidase. Again here the lysyl oxidase is inhibited and cross-linking is affected, so there will be producing a condition called laterism. Now what is a fibril? Fibrillin produces a disorder known as MARFAN syndrome, that's why we are discussing that. It's a large glycoprotein secreted by extracellular fibroblast, and it is found in the zonular fibers of the lens, that is seen there, and also in the periosteum and the arterial wall. So these are the important sites where the fibrillins are seen. Forms part of the insoluble microfibril which acts as a scaffold upon which the elastic fibers are deposited. So like that it is usually present. But a genetic disorder called MARFAN syndrome, their results are from the mutation of the fibrillin. So when the fibrillin protein is mutated, then the disorder called the MARFAN syndrome occurs. It is an autosomal dominant and characterized by ectopis lentis, or the subluxation of lens, because the zonular fibers of the lens is affected due to the mutation of the gene coding the fibrillin. Then an abnormal maturation of the skeleton and the aortic aneurysm. So when in the walls of the aorta also there is fibrillin which is not functioning properly, then aneurysm means dilatation of that part. Then there will be some abnormalities of the skeleton, and that is the Charlie Chaplin gait and everything in the homocysteinuria. So here MARFAN syndrome, it is an inherited as autosomal dominant manner. Defect is in the gene encoding the fibrillin 1. It's a glycoprotein. Fibrillin is a component of microfibrils which gives stratum for the deposition of elastin. So it forms a stratum for the deposition of elastin. So when this stratum is not there, elastin cannot be deposited. So that leads to the defective function. So deposition of fibrillin and elastin becomes low, which leads to the symptoms. That is arachnodactyly. Long digits may be there for these patients. And ectopia lens, that is dislocation of lens, hyperextension of joints, and also typically you can see the aortic aneurysm in this case. Now elastin, what is elastin? It's an extracellular matrix protein, provides the recoil to the tissue that undergoes repeated stretching. So whenever there is a tissue, it has to undergo repeated stretching. The recoiling of that tissue is done by the elastin. It is tightly associated with the collagen fibrils. The major component of elastic fibers are found in ligaments, lungs, and walls of the blood vessels like aorta. Small amount is seen in the skin and ear cartilage and the bladder. Ear cartilage and bladder also, some amount of these elastins are seen. Elasticity of the skin is mainly due to this elastin. And glycine is 1/3 of the amino acids, and proline and alanine are also seen in large amounts. So here glycine again is 1/3 of the amino acid of the elastin, then of course proline and alanine. Small amount of hydroxyproline also can be seen. Hydroxylysine is not there, so glycosylation is also absent. So usually the glycosylation occurs when there is small amount of when only to the hydroxylysine. So when there is no lysine, hydroxylysine, glycosylation also is absent. Mature elastin is very stable, and the turnover is very low. There is a condition called pseudoxanthoma elasticum. It is an inherited defect in the formation of elastin. Clinical features almost same as that of EHLERS-DANLOS syndrome. This condition is called pseudoxanthoma elasticum. Now the basic units of subunit of elastin is the tropoelastin, consists of 800 amino acids. No repeated units like glycine, proline, and hydroxyproline, but have glycine and proline in high amount. And it contains alanine and non-polar amino acids like valine, leucine, and isoleucine. And cross-linking of the elastin is by forming desmosine. The desmosine is the one which is formed in this, just like tropocollagen. Here the desmosine is helping in the cross-linking of the tropoelastin. It is derived by condensation of 3 L-lysine, that is L-lysine means oxidized lysine, with 1 lysine, normal lysine. So like that, when 4 lysines are present, there will be formation of desmosine which can help in the formation of tropoelastin and cross-linking of the tropoelastin. So the cross-linking permits elastin to stretch in two-dimensional direction. So the importance of this cross-linking is that it helps in the cross-linking of the elastin in stretching two-dimensionally. Now what is keratin? Keratin is present in the hair, skin, horn and hoof, and it consists of a matrix, keratohyalin, and the fiber is alpha keratin. The fibril has 3 polypeptide chain and 10 to 12 fibers form one bundle. Just like the collagen, there are 3 polypeptide chains together forms the fibril, and there are about 10 to 12 fibrils to form a one bundle. Keratohyalin matrix contains cysteine-rich polypeptide held together by the disulfide bond. So this matrix of the keratohyalin contains cysteine-rich polypeptide held together by the disulfide bond. More the disulfide bond, harder the keratin. seen in the hair and give the mechanical strength. So the keratohyalin is the one which produces the tensile strength to the keratin. So there, when the disulfide bond is more, then it will be more thicker. So disruption of these bonds increase the solubility and decrease the tensile strength occurs in the artificial waving of hair. So that is what happening in artificial waving, though they are disrupting the disulfide bond in the keratin. Now, another important disorder is the Duchenne Muscular dystrophy. Dystrophin is a structural protein attached to the muscle cell membrane, and it links the actin of the cell to the extracellular matrix. So that is the function of the dystrophin. It links the actin to the extracellular matrix, cell actin to the extracellular matrix. And it causes, here the cause for the DMD is mutation in the dystrophin gene, and mutation in the gene of the glycosyl transferase enzyme also leads to the some variety of DMD. So either mutation of the dystrophin gene or the gene which codes the glycosyl transferase enzyme. Absence of dystrophin allows the excess calcium. So here what happens is, when there is absence of dystrophin, it allows the calcium to penetrate into the cell through the membrane, and calcium, when it enters the through the cell, it brings the water into the mitochondria, and then the mitochondria burst. So along with calcium, water also enter and into the mitochondria, and mitochondria burst. That is the thing happening in the DMT. Now the clinical features. There will be muscle weakness, starts by 4 years, and it worsens very fast, and usually starts in the upper part of the leg, pelvis, and the upper arm, and the patient is unable to stand up. So the upper part of buttock area, that is pelvic area, where the upper part of the leg and the pelvis is there, which is helping it to stand up. Similarly, the upper arm also. So these parts are affected, and that too early, when he is 4 years old. Then most are unable to walk by 12 years. So these children are unable to walk even up to 12 years. Then the affected muscle look larger due to the increased fat. So the fat deposition occurs there, and then the muscle will be bulkier, seen as bulkier. Females are the carriers, no cure for the disease, and usually the patient dies by the age of 25. So there is no treatment for this, only the symptomatic treatment. So this much regarding the revision class, but we have to study the muscle proteins like actin, myosin, and that we have studied in detail in physiology, and that troponins, C, I, E, etc., and their functions, and also some lens proteins are there, that is lens proteins, crystallins, the main thing, alpha, beta, and gamma crystallins are there, and how the cataract is formed when there is defect in these fibers, or what are the pathogenesis of cataract in the diabetes and other conditions, in bone errors, galactosemia, and all that that you can study. Then there are one part again, prions and prion disease. That is already, I think it is discussed along with the structural organization of proteins. So that also can come sometimes, prion disease. So that you can read in your textbooks and refer back your previous notes. Okay, so I am not going to take all these things together, just revising some important point regarding collagen, and then elastin keratin, what is given in our syllabus, that part, and then I don't know what are questions going to come. So read the chapter almost thoroughly, because it is a new chapter, new topic for your new syllabus. Okay, thank you.